Hello everyone, myself Ncert from Ncert Medical College. Let's start with page number 96, Cell the Unit of Life, Ncert Biology class 11. Now continuing with the Golgi apparatus, here it is mentioned about the concave trans or maturing phase. Trans phase is also known as maturing phase. You have to remember this, and why it is called maturing we will learn later. Now the cis and trans phases organelle are entirely different. You have to remember that these are entirely different, but they are interconnected. You have to remember this terms. Now the Golgi apparatus principally functions as packaging of material. This question have asked previously that function of Golgi apparatus, it is the packaging of material. In other options the options were packaging of materials, secretion of materials, releasing of materials, synthesis of protein, etc. But you have to mark this packaging of materials. Now materials to be packed in the form of vesicles from the endoplasmic reticle fuse with the cis phase. You have to remember this because firstly there comes to the cis phase from the endoplasmic reticulum, and the Golgi apparatus move towards the Golgi apparatus and move towards the maturing phase. From cis phase to the maturing phase it moves. You have to remember this function and the movement of the material. Now it is the region for which Golgi apparatus remain attached to the endoplasmic reticulum. It can be your board question. A number of protein synthesis by the ribosomes in the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from the trans phase. You have to remember that proteins were modified in the Golgi apparatus. You have to remember this thing. Now the Golgi apparatus is an important site for formation of glycoprotein and glycorapid. You have to remember this too as well, because the formation of the glycoprotein and glycolipid has been asked previously. Now coming to the lysosomes. Lysosomes are also membrane bound vesicular structure, and they have various type of hydrolases, that is enzyme, hydrolytic enzymes, that is hydrolases, and these are lipases, proteases and carbohydrases, and they are capable of digesting carbohydrate, protein, lipid and nucleic acid. You have to remember. And the optimal pH is acidic. You have to remember this. In PET question they might ask you this question. In lysosomes the pH is acidic or basic, so you have to remember acidic pH. Now coming to the vacuoles. Vacuoles, a very important question which have been asked previously many times is that vacuole is bound by single membrane. You should not confuse it double membrane organelle. It is not a double membrane organelle. It is a single membrane organelle, and the membrane is called tonoplast. This question also have been asked previously. And the function of tonoplast can be a question can be asked in your board examination. That is, function of the tonoplast is facilitates the transfer of number of ions and other materials against the concentration gradient. You have to remember this term, against the concentration gradient, into the vacuole. Hence their concentration is significantly higher in the vacuole than in the cytoplasm. You have to remember this thing, that it is against the concentration gradient tonoplast works. Now amoeba and protist, two types of vacuoles are seen. In amoeba, contractile vacuole which helps in osmoregulation as well as excretion, and in protist, food vacuoles formed to engulfing the food particles. You have to remember these two type of vacuoles. Thank you.